Today I will be discussing the vestibular system. I will cover anatomy and physiology of the semicircular canals and the otolithic organs, afferent innervations, and the horizontal semicircular canals, vestibulo-ocular reflex, or VOR. So first, let's discuss the semicircular canals in the version of pretzels. There are three canals: the horizontal semicircular canal, anterior semicircular canal, and posterior semicircular canal. Endolymph, a substance high in potassium and low in sodium, fills these canals. Gave a fun little example of a large banana and just a tiny little bit of salt, so enjoy that pun. These canals are arranged orthogonally to one another, which means they are all at right angles to one another. This fact is not only important for understanding their anatomical structure, but also for understanding that this is what enables the system to detect direction and amplitude of head movement in any direction. After all, the semicircular canals are responsible for angular acceleration. Each semicircular canal has a contralateral coplanar mate. So, right here we have the left posterior canal, and that's mate's going to be the right anterior canal. And then of course we'll have the left anterior canal pairing with the right posterior. And then of course the horizontal canals will always be pairing with one another. So the left and the right horizontal canals. So while one side of the pair is excited, the other side is going to be inhibited. Before diving further into this excitatory versus inhibitory concept, we must discuss some more anatomy. Each canal has an enlarged end called an ampulla. So let's shift our focus to this nice little portrayal. This bowl is going to represent our ampulla, and it's going to contain a sensory organ called the crista ampullaris, and that's where our muffin will be represented. The crista ampullaris is shaped like a dome and contains stereocilia and kinocilia of each of the hair cells. Beneath the crista ampullaris is the cupula, a gelatinous mass that extends from base to apex of the ampulla and effectively serves as a barrier between the semicircular canal and the vestibule. That will be represented by our lovely Greek yogurt. Kinocilia and stereocilia extend into the cupula and are the physical structures that respond to cupular deformation. Deflection of the stereocilia toward the single kinocilia is going to represent excitation or depolarization, whereas deflection of the stereocilia away from the single kinocilia is going to lead to inhibition or hyperpolarization. So now that we understand what the ampulla is, it will make more sense to discuss the terms ampullofugal and ampullopedal. In the anterior and posterior semicircular canals, endolymph is moving toward the ampulla, and it's going to result in a decrease in firing rate in the nerve, and that's when it takes on the term ampullofugal. So just to review, that's the anterior and posterior canals. In the horizontal semicircular canals, endolymph is moving towards the ampulla, and that's going to result in an increase in firing rate in the nerve, and it'll take on the term ampullopedal. Each of the semicircular canals responds best to motion in its own plane, with coplanar pairs exhibiting a push-pull dynamic. For example, as the head is turned to the right, the hair cells in the right horizontal semicircular canal are excited, whereas the hair cells in the left horizontal semicircular canal are inhibited. Next, let's tackle the otolithic organs. There are two otolithic organs, the saccule and the utricle. The inertial mass within these otolithic organs comes from the stereocilia projecting from the tops of specialized hair cells into a gelatinous matrix that contains otoconia embedded in it. Otoconia are calcium carbonate crystals, and this gelatinous matrix is known as the otolithic membrane. For otoconia in this representation, I used Himalayan pink sea salt crystals, and they're lining right here on the utricle. I left them off the saccule just simply because they'd fall off without glue. And their presence is what increases the specific gravity above that of the endolymph. This is why the otolithic organs are responsible for linear acceleration. And just as a reminder, the semicircular canals were responsible for angular acceleration. So within each otolithic organ is an anatomical midline of otoconia that divides the organ into two parts. This is known as the streola. The polarity patterns of hair cells orientation to streola enables an organism to detect linear head motion in nearly any direction. The kinocilia of utricular hair cells, that's going to be seen by these arrows, are oriented toward their streola, this midline, and the kinocilia of the saccular hair cells are going to be oriented away from their streola. Utricular excitation happens during horizontal linear acceleration, and saccular acceleration happens during vertical. Now we will discuss vestibular afferent projections. Each of the end organs in the peripheral vestibular system sends their dendrites to the cell bodies located in Scarpa's ganglion. This is the origin of two branches of the vestibular nerve that are located in the internal auditory canal near the emergence of the vestibular nerve into the cerebellopontine angle. These two branches include the superior vestibular nerve and the inferior vestibular nerve. The superior will innervate the horizontal and anterior semicircular canals, utricle, and small portion of the saccule, while the inferior vestibular nerve will innervate the posterior semicircular canal and the inferior division of the saccule. These two nerves run together as a bundle into the pontomedullary junction where they bifurcate. Primary vestibular afferents in the superior branch include axons that synapse in the superior or medial vestibular nuclei of the uvula, nodulus, flocculus, or fastigial nucleus of the cerebellum. Primary vestibular afferents in the inferior branch include axons that synapse in the medial, lateral, or inferior vestibular nuclei, which along with the superior vestibular nuclei and other subnuclei comprise the vestibular nuclear complex. A practical way of looking at this includes afferent projections from the ampullas of canals that project to vestibular nuclear complex and then connect to motor neurons of the extraocular muscles. Because of each extraocular muscle receiving both excitatory and inhibitory input from the semicircular canal system, the eyes move in approximately the same plane as the semicircular canals. And that's a perfect transition for our final topic, the VOR. In our last segment, we will be discussing the horizontal semicircular canal vestibulo-ocular reflex, but first we must unpack the extraocular muscles. So there's going to be 6 extraocular muscles for each eye. In this example, this tangerine will be our right eye, and this one will be our left. So we'll start with the medial rectus right here, the lateral rectus, superior rectus, superior oblique, and inferior oblique. For each eye, these 6 muscles each have opposing counterparts comprising of 3 different pairs. These pairs will include medial rectus and lateral rectus, superior and inferior rectus, and the obliques. Each muscle in a pair moves the eye in the same plane, but in the opposite direction. So the agonist muscle is going to be pulling the eye in the direction of interest, and the antagonist muscle is relaxing. This is oversimplified, of course, but it's going to be helpful in our understanding. So we'll note that the medial and lateral recti are responsible for adduction and abduction, or horizontal movement. The superior and inferior recti are responsible for elevation and depression, or vertical movement. And lastly, our superior and inferior obliques are responsible for both vertical and torsional movement. Specifically, the superior producing depression and torsion, and the inferior producing elevation and extorsion. So now moving on to the cranial nerves and oculomotor system nuclei. Extraocular muscles are innervated by oculomotor neurons located on each side of the brain. The third nucleus, or oculomotor cranial nerve, innervates ipsilateral medial rectus. We're still going to be talking about the right as our priority for ipsilateral, and the left will be our contralateral. And I pointed to ipsilateral medial rectus. Then we'll have the inferior rectus, the inferior oblique, I'm blocking it, inferior oblique, and the contralateral superior oblique. Then we will have the fourth nucleus, or the trochlear nerve, and that's going to innervate the contralateral superior oblique. The sixth nucleus, the abducens nucleus, cranial nerve, will innervate the ipsilateral lateral rectus muscles. Oculomotor neurons control the velocity and position of the eye by continually adjusting the neural input to the extraocular muscles. Now we are ready to discuss the horizontal semicircular canal VOR 3-neuron arcs. First, we will discuss the excitatory 3-neuron arcs. Angular acceleration and deceleration of the head are what activates the VOR, so let's turn our head to the right in this example. This grape will be our right eye, and this one will be our left. Starting with reflex arc 1, we will be following the goldfish. Primary vestibular afferents from the horizontal semicircular canal synapse in the ipsilateral medial vestibular nuclei. The right medial vestibular nuclei projects the increased level of neural activity to the contralateral abducens nucleus. Motor neurons from the left abducens nucleus then synapse at the lateral rectus muscle of the left eye. This results in eye movement in opposite directions of head movement with equal velocity. Moving on to reflex arc 2, we'll be following the Cheerios. Primary vestibular afferents from the horizontal semicircular canal synapse in the ipsilateral superior vestibular nuclei. The right superior vestibular nuclei projects this increased level of neural activity to the ipsilateral oculomotor nucleus via the ascending tract of Dieters. Motor neurons from the right oculomotor nucleus then synapse at the medial rectus muscle of the right eye. This too will result in eye movement in opposite direction of head movement with equal velocity. Lastly, the reflex arc 3 will be the chocolate chips, and that's going to be motor neurons from left abducens nucleus crossing the midline via the medial longitudinal fasciculus to the right ipsilateral oculomotor nucleus, and then they're going to synapse at the medial rectus muscle of the right eye. This results also in eye movement opposite to head movement with equal velocity. And just to wrap up, there is an inhibitory 3-neuron arc. It's going to be the exact same pathway, but everything will be in reverse. So we'll be starting with the left horizontal semicircular canal. This inhibitory pathway provides relaxed muscle response to facilitate opposing contraction of eyes in the opposite direction.